I also want to welcome everyone who's joined us. So, Jack Johnson is a mycologist from the Pacific Northwest. He's currently pursuing his master's degree at the University of Wisconsin-La Crosse in the lab of Dr. Todd Osmondson. Jack has conducted field research across the United States, Guyana, and Trinidad and Tobago. His current research focuses on the biodiversity and biogeography of the Hawaiian fungi. So welcome, Jack, and thank you so much for not only putting this presentation together, but making it specific for us here in the Midwest, which I really appreciate.
Yeah, for sure. If I could be granted sharing.
Yes, you are now a co-host.
Excellent. Moving around the buttons. Bing, bang, boom. Oh, what did I do? Okay, here we go. Making it tiny. I'm moving the zoom options, so I don't know if you can see me struggling to do that, but that's my current endeavor.
Hey, look, it's your mom at the old cabin. Do you see that?
Okay, so thank you for tuning in. My name's Jack Johnson. Hopefully you have heard this talk title and you know what you're in for, but don't worry, I have considered that there are differing interests in this group of people. So if you haven't, it's suited for those who weren't expecting to be dosed with strange fungus as well. So yeah, let's get into it. A little bit about me. All of these pictures are me except for this one here, which you might have noticed. So I give talks. Right now, I'm a master's student in the lab of Dr. Todd Osmondson, who is an instructor, professor at University of Wisconsin-La Crosse. I'm studying fungi of Hawaii, as Mariah mentioned. And before that, I worked doing Myco Club nonprofit stuff up in the Pacific Northwest. I had a job touring around the country and doing bioprospecting. So I was looking to do soil sampling of interesting fungi for a company that was doing drug discovery. And that was pretty neat. But I love sharing my hobby slash career with the general public. It's fun. You know, it's great. We have this community of mycological minded people to share it all with. That being said, I do imagine there's some differing interests in this audience. So if you are any one of these three, I imagine there are some mycophiles here, huge nerds. You're going to be happy to see pictures of strange fungus. That's what you want to do. You do it whether or not you're in this presentation. So there's stuff in here for you. There's the generally curious in this group, those among you who are naturalists, are more casually interested in mycology. You're a member of this club and you're trying to learn more. This will be approachable. We're not going to get too out in the weeds. It'll be new ways for you to find fungi in your habitats, your local areas. And then there are those of you who figured, they just wanted to put something on other than Netflix while they're eating dinner. And for those of you who are in that bunch, there are pretty pictures. And this is a presentation where if you dip out for a few minutes, you won't be totally lost in the sauce. So don't hesitate. Some presentations, you look away for a minute and you're lost. This one is not that. So the precedents for this talk, I'm not talking to you necessarily about uncommon fungi. I am talking to you about cryptic fungi. So ones that dwell in atypical places. They're not the casual log rotters that we know and love, but that does not mean they're uncommon. So in studies that look into inventorying biodiversity of plants, people review those studies to try and find out, okay, well, what kind of methods worked? What are the errors? Why do we miss species? And I'll tell you why we miss those species in a second, but I'm also going to talk to you about small, visible fruiting structures. And that doesn't mean they're ecologically insignificant. So just because a little bump on a log that I might show you is to the naked eye small, remember we're talking about fruiting bodies or fruiting structures. and you can have a very small fruiting body, but have a fungus, the mycelial body corresponding, that could be large, could be filling a large role in an ecosystem. And if you see a star at the bottom left of the slide, that means it's something that's found in the Midwest. So it's one that you can get out and look for here. I am not here right now. Here being Wisconsin, I am in Seattle from my little cousin's bedroom, if you were wondering. So observing less obvious organisms is crucial for us to actually have an idea of what biodiversity exists. It's also a good time. And so back to me talking about surveying for plants. When I read there's a review article that I was really interested in, looking over 59 different studies of surveying methods. It was called Observer Error in Vegetation Surveys, and it's a big review paper. And so most all of these studies found some type of human error, one or another, misidentifying things, overlooking things. And what I want to point out, and the value of learning about these cryptic niches, is we get hung up on putting the right identification on things. It's so much of what we do when we sit and blab around names, or maybe we're past that and we go, we'll just put a code name on it for now, and that's good. But only 5 to 10% of these studies showed misidentification errors were significant. Meanwhile, 3 to 6 times as much, so 10 to 30% of errors in these identifying plants in a given habitat were just due to overlooking them. They use a fancy term, pseudo turnover. But between surveyors, if I have one botanist go to a forest, inventory the plants, and I have another one, misidentification, not that huge of an issue compared to just plain old not seeing it. And in mycology, there's lots of fungi that I didn't see, didn't know how to find until someone showed me how to find them. And that's what I'm hoping to accomplish by sharing some interesting things with y'all. New ways to look. So yeah, overlooking 3 to 6 times more prevalent than misidentification. And also, this study talked a lot about having people with different interests being really important. For instance, if we have someone who studies Russula in our club, you're going to get more species names on your Russula. And if you have a foray where Tavis isn't there, you're going to have less of those names. Having people who can study crust fungi is important. And crust fungi, depending on survey lists you look at, they can make up a majority of macrofungal diversity. And I want to call out right away, this is a bias, sort of a biased thing to try to, it's really hard to get rid of bias when you're trying to say crust fungi can make up a majority of the fungal diversity. They're biased by persistence, so the crusty structures stick around for a long time. And they're also biased, if you're this, to get this number, they had to have general mycologists going out trying to find everything. Because if you're a bunch of crust and polypore loving people, so crust fungi, I'm talking about flat things on logs or on soil, you're going to find more of them, right? And I didn't ask Andrew's permission, so pardon me if this was not the thing, but we have a crust and polypore lover in our club, Andrew Kitson. And in this study that studied a spruce forest, 75% of their species, which was only 60 species, were crusty things. So not only can we fill in some gaps by looking at the less glamorous fungi, but sometimes we're really missing a majority of the diversity if we don't look at them. And so for this talk, we're going to focus on macrofungi, stuff you can see with your naked eye. But I do want to point out really quick some of the beautiful stuff that you can see when you zoom in. So this is a crust related to Cantharellus. It's in the same order as chanterelles. It's called Botryobasidium. And I think it's just a beautiful happenstance of nature that things that look like, maybe cat barf on a log under the microscope, fantastically gorgeous, strange little things. Creek foam. Here is a pile of creek foam that I waded into. And this picture's not mine, but there are gorgeous spores of Ascomycota and others hanging out in the creek foam. That's an unusual niche that you can observe in your own area. And you want the foam to be really pure white if you're going to do it. I've sampled creek foam most every time I've been able to, if I had a microscope. And the darker and cruddier looking the foam, the less quality, the less easy it is to find these, and you just find a bunch of debris. And golden little powder on a log. This is Maricel Patino's photo, a wonderful, wonderful mycologist. This golden powder under the microscope is a genus called Helicosporium, and helix shaped, gorgeous spores. So there's a lot more that we'll not touch on today, but I'm going to focus on some more charismatic looking things or more unique things. But I encourage you to stare at crusts on logs. It's good for your health. And so jumping in, the first kind of category I want to introduce you to are fungi that mimic flowers. They get called fungal pseudoflowers, and there are a few different lineages that have come to this ecological situation. 3 different in specific. There are the rust lineage, and then there are 2 Ascomycota lineages. So these fungi play a trick on pollinators, essentially, where to be considered a pseudoflower, you are mimicking some sort of smell or signal for pollinators or just general insects, maybe not bees all the time, to come by and spread your spores or spread your hyphae. And this, again, 3 different lineages, so it's convergently evolved. This isn't one ancestor gave birth to all of the fungal pseudoflowers. It happened 3 times, which is pretty neat. So I'm going to give you a before and after. If you're thinking, wow, that's a fungal pseudoflower, it looks so much like a flower. Yeah, that's just a flower. I'll show you the fungal pseudoflower on the next slide. So hold your eyes on the left. We're looking at a species of Xyris in the yellow-eyed grass family. And these are photos from Guyana when I was doing field work with the AIM lab and some people from Penn State. We were trying to study what's the deal with the fungal pseudoflowers that grow on these Xyris. So hold your eyes on the left one and see this tissue? That's all fungal. It's really tempting to think, oh, it transformed the petals, right? There's not plant tissue in this. This is all fungal growth. And we were studying while we were there the volatiles, so the smells that these fungal pseudoflowers are letting out. And we were observing what insects came by. We set up little GoPros and observing what insects came by and visited them. What's tricky is you think, okay, so maybe this is spreading spores, right? No, in this instance, this is vegetative. There's not conidia. There's not spores, asexual spores being formed in here. And instead, at this point, I don't know that it's proven how this fungus spreads, but instead, we saw a lot of grasshoppers come by and eat the tops of these, and it's possible that the vegetative hyphae are being spread through insect vectors. But who would have thunk a fungus mimicking a plant? This one's not in the Midwest, but there are other fungal pseudoflowers that are. So here's a host of a different fungal pseudoflower. This is a Bochara, or I think maybe they're called stone crops, the common name. This plant's in the Midwest. This genus is in the Midwest. I spent some time looking for the fungal pseudoflower. But this one's interesting. The fungal pseudoflower that infects it, in our last example, it manipulates the area where the flower is produced, right? In this example, there is a rust fungus that moves in and causes leaves to be clumped up and become bright yellow. And instead of changing a flower, it changes the leaves to look flower-like. And I saw this in California for the first time. This was described by a mycologist who's done a lot of work with pseudoflowers named Biddy Roy. Biddy rules. Really cool researcher. And I even saw a golden crab spider hiding on these yellow pseudoflowers. So they are convincing enough to where not only do they attract insect vectors for pollination, but these visual cued spiders that like to dwell on yellow flowers set up shop and were waiting to catch some of the pollinators, which is beautiful, beautiful. So yeah, this is related to rust fungus that you might be familiar with, the ones that grow on your apples or your cherry plants or the junipers in town. But what a strategy. What a strategy for living. Another fungal pseudoflower that is, again, not manipulating. Well, this is two-part. It's manipulating the flower and it's manipulating the foliage, the leaves, to have its ecological role be fulfilled. So Monolinia vaccinicorymbosa, they call them mummy berries. People call them mummy berries. I had a friend taste one once. They don't taste great, if that was a question that came into your head. They taste bad, and maybe like nothing too bad is what was reported. So this fungus infects blueberries. I'm not sure if it infects other huckleberries and other relatives, but definitely a problem in the blueberry industry, and it's a common thing you see out in the woods. And it does something interesting where the flowers are white to pink, and then after the flowers have gone, it makes a white mass out of the berries, which it's interesting that it's similarly colored to the flower. And then it produces uh, a volatile, some sticky sweet stuff on the leaves. And what's been found is that the transmission of the monolinia, of the pseudoflower fungus, actually happens by bees being attracted to the sticky smells and stinky smells that it produces. And from a seasonal perspective, it's kind of interesting to think, uh, these bees, this is, perhaps at the end of their season, the berries have the flowers and the pollen they want have come and gone, and we're at the time where there's more berries, although there are still some flowers you can see in the background here. But uh, perhaps also it plays into the phenology, the timing of the year, and is producing another source of sugar sweetness when there are less to be had. So this is one you can find if you go out in your woods. Well, I like giving this lecture in person, especially where there are blueberries nearby. Uh, at one time I gave this presentation, 2 of the fungi I mentioned were found within 50 feet of the presenting area afterwards, which is pretty neat. So another flavor of plant interactions are those fungi that grow on anthers. So the structures that produce the pollen, there are fungi that will infect them and make a powdery mess. And again, pollinators come and land and they carry away the spores of these fungi. So this one, Anthrospora scilla, grows on relatives of hyacinth, I believe, in the bluebell world of plants. And I've got a question mark next to my star because I don't believe this one's been found in the Midwest, but I also don't think anyone's really looking that hard. If anyone knows if it's been found, pop in the comments so we can have some updates. But this is another example. If you know this is there, you could be the first person to find this in your region, which there's an excitement there, right? To be able to be contributing to our understanding of biodiversity. So if you ever zoom in and you see these big fluffy anthers, that could be a anthericolous, an anther-loving fungus. There's another one that grows on chickweed, and I believe chickweed just had a genus name change, so it's named after the former genus of chickweeds. And I think, so I put a star here, so there's records that this has been found in the area. Again, it's making these totally strange structures out of the anthers. And the first time I gave this on the West Coast, gave this talk, uh, it had not been found on the West Coast. And now there's a handful of records and sites, and turns out just telling people about it's the biggest barrier for knowing it exists in a region. Now we know it's in California. There are researchers who are studying Mycobotrium as a proxy, as a way to understand smut fungi that grow on our corn. If I have that right. There's a related plant pathogen that's of commercial interest, and there's a lab that does work on Mycobotrium to see if they can learn more about the plant, that pathogen's relative, to be able to inform better practices to save our corn, because goodness knows we need our corn. So another place on plants that you can look for fungi that you might not have thought to is on the resin, especially with conifers. So up in northern Wisconsin, if you are the kind of person who is willing, spend a day looking at sap, little pockets of sap where it's maybe dried and run a little bit. And you can find little cup fungi. This is Lachnolula resinaria. And there is a whole bunch more to be found. There's brilliant purple ones. There's all sorts of different flavors of resinicolous fungi. And without people looking for them, we probably won't find any more. So please be the ones, spread the word. These fungi, it's interesting to think, plant defenses to keep fungi out. One of the good ways to keep fungi from infecting your wounds, right, is to produce a whole bunch of resin that are full of volatile organic compounds. I don't know the history, but at some point, some ascomycota, some cup fungi, figured out how to grow on this resin, and they made their keep. So there is no unfilled niche in nature, right? And if you can be the first one to learn how to eat pitch sap, you've got it made. So get out there and look. This is another one. This is an asexual form of a ascomycotin that grows on pitch, but it will also stick around after the pitch is gone. You might see some big conifers up in the northern part of the state and see a black spot and think it was burned. And that's what I thought for years until I had someone told me, look closer. Sometimes those burned patches are actually a species of fungus. And up close, there are these crazy networks of cinnamata, these structures that form little asexual spores. They spread some spores through it. And I think it would make a great cover for a heavy metal album. So if that's something you're trying to find an album cover for right now, here's a candidate. And something I've noticed, at least in the Pacific Northwest where I'm from, is when you find these fungi, and if you know they're in an area you like to naturalize or walk, you can kind of use them as a metric to understand how much it's rained in that area recently. So while some, like in the middle, there'll be like more of these cord-like growths, the powdery black cinnamona and some of the powdery stuff on the base washes away with the rain. So I can, when I was naturalizing in drought times in the Pacific Northwest, I can walk from area to area in my forest, and where those patches are bigger and bigger, I know it's likely to have rained less because they haven't washed away. So it's an interesting way to gauge, learn something about where you're at too. Also, if anyone has any questions, you can pop them in the chat. If you have something pressing or you'll think you'll forget it, you can interrupt too. This is casual. So myxomycetes are slime molds, and we're moving on. We're moving away from the strict plant-associated ones. We're going to talk about fungi eating other things, existing on other things. Slime molds have a species of fungus that grows on them called Polycephalomyces tomentosus, and they'll form these wacky little structures that produce conidia. And in our area, you can find them on Leocarpus fragilis, the pretzel slime mold, and they're really pretty small. So this is kind of a fungus that typically you're looking closely at a log, looking at wet, damp undersides of logs, maybe near the soil where there's been some moisture and coolness locked in. That's where you go to find slime molds most of the time. And if you see little white specks growing on the already tiny slime mold, that might be Polycephalomyces tomentosus. If you are a lazy naturalist and you really want to see this, or maybe a patient naturalist, another alternative, and the way I found it for the first time, is to collect the slime molds and then leave them on your desk for a few months. And afterwards, sometimes you'll see the polycephalomyces creep out of them, which is pretty dang neat. Another myxomycete muncher, a slime mold muncher, is Nectriopsis violacea. It's related in the same world as our lobster mushroom, and instead of infecting something and turning it bright orange, it infects Fuligo septica and turns it purple, which is really, really cool. This one I see, I tend to see it on well-rotted standing trees most of the time that I find it. Um, this is a really nice specimen. And so what we're looking at is a felty subiculum-like structure. So this mat that it made, and these are little or parathecia, which are take your cup fungus, close it up into a little ball, and spores are being shot out. And now you can see they've deflated after they've shot out their spores. Uh, this one has been found in the region. You can get out there and find it. What you're going to do is you're going to look for um, Fulgoseptica, which is this dog vomit slime mold, and look for Fulgoseptica in shadier patches, especially on older rotted wood is where I've seen it the most. I've only seen this one less than 10 times, but if you see a bright purple blob, now you know. We got Nectriopsis violacea in this beautiful world of ours. So these are fungi parasitizing or eating the remains of slime molds, which are amoebozoa. They're not fungi, right? We used to think they were. That's why we lump them in being molds. They're like that cousin that you call family now, even though you know they aren't really your family. And so there are ones that predate on plants and plant parts we've seen. There are fungi that predate on myxomycetes, slime molds. And then there are some that predate on mosses, so a different lineage of plants. In the Pacific Northwest, I've spent a lot of my spare time looking around sphagnum patches. So sphagnum it's a very heavy metal lifestyle. Basically, the dead growth of moss turns into a sponge, and then they keep growing. The sphagnum moss keeps growing on top of this spongy mat of its dead ancestors. Kind of like the soft land version of coral, where coral are growing on the dead exoskeleton of their previous relatives. So in that moss, there will be fungi that you don't see anywhere else, and some of which have been assessed for conservation. They are thought to be rare. The truth is, I think we just need a bunch of people looking at sphagnum before we can really know. There's one that's listed that was found for the first time in either northern Minnesota or northern Wisconsin or around Minnesota, Des Armillaria ectopoides. So there's like a honey mushroom relative that you can see. That one's listed as vulnerable, I believe, and it was the first time it was seen in North America, which is pretty cool. What we've got here are some waxy caps. We've got Hygrosope turunda, which is there's some similar looking ones, but what you're going to want to look for are black scales on the cap that kind of darken, especially as you dry it out, they darken. They can be upturned like this, where the cap margin goes up and out. And when they're younger, they can be a little bit more down facing, and they've got like kind of this scalloped gill margin. And then we have Cuphophyllus cinerelis, which is a really beautiful species. I might have forgotten to take this out. I don't know if Cuphophyllus cinerelis is found in the Midwest, but there are a whole bunch of other sphagnum fungi. And so you're going to want to look for big continuous patches of sphagnum to have the best chance. And just some mindfulness when you're in these habitats. These are habitats that have been incentivized to be destroyed during the Swamp Act. Early in our country's history, or earlier, I think in the early 1900s, people were paid to destroy sphagnum. Sphagnum is mined. Wetlands were thought to be these areas where there's lots of disease. So if you go to a sphagnum patch, wear high boots. 1, don't bring anything you want to get really wet, or that you don't want to get wet. 2, and 3, when you're walking, don't stand and in one place for too long. You can sink and kind of rip things apart. Don't jump. Be gentle with the sphagnum, because I don't remember if it's a millimeter every 10 years, but they can be big mats, but they're deposited very slowly. So get to some sphagnum near you for some unexpected fungal treats. This one is a not glorious picture, but it's one that I found at our Perot State Foray a year ago. It's in the same group, Eochronartium muscicola. It's in the same group as the rust fungi, and it likes growing on moss. And I thought these might have been some mushroom that lost their cap at first, but it turns out it's its own dang thing. And I don't know how well reported it is in the region, but it's definitely one to keep an eye out for. It was growing on pretty exposed moss, just on a fallen oak in the, I guess that's beginning middle of spring, and they just form these thin little threads, and these are only, a centimeter high or so. So keep an eye out. It'd be nice to know more of where this grows, and take good pictures of the moss. It'd be nice to know what moss it's growing with.
We're talking, everybody can hear me.
So now we're going to move into fungiculous fungi. So those which munch other fungi. So we've got, to start off, the body snatchers, Squamanita. So Squamanita is in the family Squamanitaceae, which is really good to say. I think if you're looking for another word to replace a curse word with, if you're around children, Squamanita, you could try that out. Might work for you. And what they do is they actually infect and grow up through the mushrooms of another species. So I believe I always confuse cystoderma and cystodermella, but you don't care. Or if you do, whatever. It's growing on another member of the same family. Cystoderma, I believe, is Squamanitaceae. And this mushroom here, this is Squamanita. This is the remnants of that cystoderma or cystodermella, and it's popping right up through it. And so there are a few species. I've never seen this. This is a bucket lister. This is a once in a lifetime thing, so far as I understand. And they grow on cystoderma or cystodermella, whichever one it really is. And they also grow on Cuernomyces, I believe, which is a clustered, maybe they called woodshank tuftlets, a clustered brown spored mushroom. And I've got a question mark. I could not find records for the Midwest. I'm not sure that there are any. I didn't exhaustively look, but keep an eye out. If you see something that looks like this, please document it. Be great to know if we could find some Squamanita in the Midwest. But what a weird zombie. This is like Alien, the popping out of your chest organisms here. Oofta. Here's another one that grows on scurfy twiglets. It grows on tubaria, the twiglets. So here's unparasitized tubaria. And hypomyces, again, this is the same genus as the lobster mushroom. I found this in the winter near University of Wisconsin-La Crosse, and I had been wanting to see Hypomyces tubaricola because I knew it existed in California, but I'd never seen it in my Californian naturalizing. But this looked like it had some infection going on, so I left it in my fridge and I came back. This was very intentional incubating. I forgot about it for about 4 or 5 days. And when I came back, it had fully infected the cap and turned it into this interesting white mass, this cauliflower-looking thing. And this is only the second record in Wisconsin, another one found by John Plischke years ago. But it just goes to show you, this might not be uncommon at all. Maybe we just don't have mycologists going out in the winter and flipping over leaves, So I highly encourage people to get out on winter walks and flip over leaf duff. There's lots of instances where interesting things have been found that way. So if you see your scurfy twiglet, which can grow in wood chips, can grow very kind of a generalist decomposer. The species on those, there's some that hybridize. It's kind of a hot mess, but you find your twiglets, you see one that looks like this, put it in your fridge. It might turn into a white blob and you might have your Hypomyces tubaricola. Another one that I found in La Crosse is a mold that keys out to, and I want to emphasize it, using microscopy, it keys out to Hypomyces mycophilus. I don't know if that's the right name, but that's what the old school keys have gotten me to. So we're going to be doing some nanopore runs in the Osmondson lab this summer, and we'll find out if the name matches the key. But on your old dryad saddle are a habitat on their own. It blew me away as a Pacific Northwester. There are so many species of fungi that grow on old rotting dryad saddle. One of them, if you see a big powdery mold, is likely what keys out to Hypomyces mycophilus. So this one, it'll sometimes in culture, it'll turn a little bit yellowish over time as well. And up here, we've got the microscopic image of the conidiophore, the structure that produces these little chicken nugget shaped things called conidia. They look like a pill. When they're fully developed, they'll have this line in the middle. And if you're a microscope savvy person, that's a way to know that you have a hypomyces in its asexual form. There's another kind of spore they can make, but and then to distinguish this one from hypomyces polyporicola, we're just measuring how big these little pills are. That's all the science really is. And so the measurements I got consistently pointed me towards Mycophilus. But keep an eye out. Another thing you can do in the winter, if you're bored and you want to naturalize, go pick up a rotten, old, stinky dryad saddle and have some fun. Asterophora lycoprodoides on Russula, blackening Russula. So there are species of Russula, brittle caps, right, that instead of rotting in a normal way, they turn into these black masses. And if no one had told you to look for them, they kind of look maybe like bear poop or something at a first glance. There's these black blobs. But if you find them, turn them over. There's some really cool fungi that can grow on them. In this instance, I've found Asterophora in Minnesota so far. I haven't found it in Wisconsin myself, but it's around. And what we have here is a mushroom that's doing a double whammy. We think of mushrooms producing their spores on the underside. This one is growing up and producing chlamydospores, so a type of spore out of the cap, and it's also producing spores on the underside. And it's an interesting trend. There's multiple fungi that grow on these blackening Russula that have two types of spore producer. They produce two types of spores on a single mushroom. And why that is has been the conversation of many excited fireside myco meetups. And I think the answer right now is nobody knows, but we've got some guesses. Another one you can see on those blackening Russula is Dendrocolibia. Dendrocolibia racemosa is a species that can form these wild, wild clusters, and they grow out of sclerotia. So they grow out of a thing that looks like a teeny peanut, and that's like their wintering structure. It's like kind of the fungal equivalent of a tuber, you know, a ball of energy to last it out. And this picture by Christian Schwarz, these photos show really well. We've got these little spindly structures coming off the side. This is where some asexual spores are being made. And then we have our typical cap and gill mushrooms. You will find it without the cap or the cap barely being formed pretty frequently. I haven't found it with this nice of cap and asexual producing structures very frequently. But so you're walking in your forests, usually a conifer forests up north, and you're flipping over black blobs, and you might get lucky and find something beautiful like this. And I've got a question mark. I don't know that it's been found in the Midwest. I think this has been put on the Northeast Fundus Rare Species Challenge. So there's reason to believe it's in the Northeast. And I don't see why this one wouldn't be in the Midwest. So get out there, flip over some black blobs. Ooh, here's a strange fungiculus fungus. Here's a strange example. So we've got our earth tongues, right? We've got these little black spoon-like things, little clubs growing out of the ground. And if you zoom way in, I want to credit before I show you, Ethan Krenson documented and shared this, and it helped get this species published. Zoom in on these earth tongues, and you'll see these little brown boogers coating it. Each one of those could be considered its own little mushroom. To my knowledge, I guess they're each their own little parathecia, their own little spore-producing structure. And what we have here is a thing called Gelatinopsis farlowii. And it grows on Trichoglossum. Trichoglossum is an earth tongue that produces these interesting little black spikes. And you can't see, this is a hand lens endeavor. Trichoglossum, I find it in kind of a few different habitats. You'll find Trichoglossum in your sphagnum bogs. You'll find Trichoglossum on nutrient-poor slopes under Cupressaceae sometimes. I also find it in really wet, low, temperate forests. Personally, I almost always find it near conifer forests, but that's where I've done most of my work. So look for that Trichoglossum, zoom in, and you might see these little bumps. And the Gelatinopsis farlowii, named for the Harvard Farlow herbarium where it was described, they root around these setae, these hairs, and they do their business. What a life cycle. How small and how wild that these things are out there to find. This picture on the left is the time I had convinced myself I had found it. I've looked for this for years now, and I think these little black blobs might have been gelatinopsis, and they're real old. But here's a healthy trichoglossum, and here's one a little more of these brown, warm tones. You can see that at a distance, but the actual structures, you got to zoom in closer to see. So what a world. We've got fungi on fungi in a bunch of different flavors. I'm not going to go too deep into these because I don't know about them, but are lichens that grow all over the place. Everywhere we've got lichens. You leave a telephone pole out, lichens will find it and start growing on them. There are fungi that predate on lichens. So these little black spots are from Tremella caloplaca. It's growing on a caloplaca, which is a genus of lichen. And this is just one of many flavors. There are in our woods in the Midwest, there are little pink fungi, and I can't remember the genus. There's two genera actually. I can't remember what they are.
Iliosporum.
What's that?
Isn't it like Iliosporum or something?
That sounds right. I'll trust
I'll look it
Mariah. So look on, and is it on Hypogymnia, Mariah?
Don't know.
If you see flat, loby lichens, little flat, sort of leafy lichens on your sticks in your forest, keep an eye out for little pink dots, and you might find yourself some lichenicolous fungi in the Midwest. Definitely a lot to talk about here, but I am not the one who knows about them, so I'm not going to talk about them.
Ah, Iliosporiopsis.
Eliosporiopsis. Beautiful.
Yeah,
Thank you. It's a very pleasing cotton candy color of pink, too.
it is.
So we're going to move into fungi that parasitize insects, entomopathogenic fungi. And I'm going to start off with a plot twist. Here's a fungus that grows on a truffle. The relatives of this fungus are all entomopathogenic, or many are entomopathogenic. However, this one in particular grows on a truffle. This truffle it grows on, Elaphomyces, is actually more closely related to bread molds, penicillium, and such than it is other truffles. And you can find a little club, so this kind of looks like our trichoglossum from earlier, that has a whole bunch of little spore-producing structures. And what you'll see with your naked eye is just where this picture tops off, right here. So I took this in Pennsylvania in the Allegheny National Forest, which is gorgeous. If you ever want to go to Pennsylvania, highly recommend. Seems like a well-kept secret, at least to me. And you'll see these sticking out. And then if you're a madman who spends 40 minutes digging to take a picture in the middle of the night, you dig down and it's attached to that truffle growing underground. Beautiful, really cool. The first time I found it, I didn't know what it was, and so I didn't dig and find the truffle. So if you find something that looks like this, dig around a little bit. There are different colors and different types of Tolipocladium, and there are yellow ones, there are black ones. I think this one is Ophioglossioides. For the sake of this presentation, you should know there are different colors. Look out for them, and if you find them, make sure you dig. So some other entomopathogens. Oh, I'm polite. Look at this. How are your brains? You've had me talking to you about random, interesting fungi for a moment now. You can take a stretch, wiggle, rub your frontal lobes. Hope your brains are all right. We're getting towards the end of this ramble of pretty pictures. So there's your check-in. Hope you're doing okay. I'm not going to stop though if you aren't. Okay, Gibellula. We've got there's a couple of fungi actually that grow on old spiders, and they make these wild structures that pop out. Some of them, this might actually not be Gibellula. I've been corrected on my ID on this a handful of times. Could be a different genus, but these are again in the Hypocreales, a group of fungi where related to our lobster mushroom world. and they parasitize tiny, tiny spiders. And in the Pacific Northwest, in my mycology club, we thought they were rare for a long time. Turns out nobody knew how to find them. They're so common that at a mushroom show I was identifying at, we didn't have Gibellula on our species list, but we had a whole bunch of plants on the table, and we had some ferns, and I just went around flipping the ferns on the ID tables, and sure enough, Gibellula was growing on a spider on one of those ferns. So a way you can find them up in ferny forests is by flipping over the ferns as you're walking. Just flip them over and look for little white dots. These are very small spiders, less than half a centimeter in size. And whoops, I clicked to admit someone on accident and then let it go. These are small spiders, and if you find one and you're the kind of person who wants to have a little terrarium sort of deal, they'll keep growing if you put them in a little tackle box. So if they aren't too spectacular looking, keep them in your tackle box for a day and they'll keep on growing. They'll grow for quite a while. My friend Connor Dooley taught a whole bunch of us how to find these, and now we know they're very common in the Pacific Northwest. I anticipate they're common in the Midwest, and if we have people looking, we'll find out if that's true. Gorgeous, strange. Last of Us is on the public eye right now, and so we have more reasons to think people want to learn about cordyceps, which is great. Here's a different flavor of entomopathogen. These are in a group that is not super closely related to the other cordyceps. It's in the Labule beniales, and these are essentially beetle STDs. This Hesperomyces harmoniae grows on our lady beetles that are now widespread and introduced, maybe invasive, I don't know. And I took this at a rest stop in Kentucky, and you can see each one of these little strands, each one of them is its own little fruiting body, and it's attached to the lady beetle. Doesn't seem to do much harm. They're kind of ecologically a mystery as far as how are they deriving their nutrients if we don't really see them sucking stuff out of the bugs and hurting the bugs that much. The only way that, from what I know, I'm not an expert in these, the only way they encumber or inhibit the bugs is if there's so many that it actually makes it hard for them to move around. So you can see these in the summertime. I would look at ladybugs flying around right now. They'll persist throughout your normal mushroom season, and sometimes even when it's hot and dry, this is a thing you can find. Very intimate shot here. So if there are children watching, sorry to bring up that conversation so early for you guys and your families. But yes, Hesperomyces. There are also click beetle family. If you are an insect person, the click beetles, Elateridae, have a lot of these. My lab mate Michael Malone is in love with Labule beniales, and if you're really into it and you want to ask someone who knows some stuff about it, we have a mycologist in La Crosse who would be excited to talk about Labule beniales with you. And then here we are. We had fungi on insects. We had fungi on fungi. There are parasites of the cordyceps. So cordyceps, in this instance, is growing on a fly. You can see a fly eyeball down here. Here, and it's covered in this powdery mass. So there are hyperparasites, a fungus on a fly. This one I found in Trinidad and Tobago. I asked the entomologists and herpetologists we were with if they knew where cordyceps grow, and they said, "Oh yeah, sure, come on." And they walked me over to a tree, as one would do, like walking someone to the aisle of a grocery store, just very comfortable. And they lifted up for some leaves and they found it instantly. And I said, "How did you know it was there?" And in Trinidad, they said, "Well, this is how high these flies land when they're going to be infected with the cordyceps. It's always that high." So some pretty specific control of the cordyceps manipulating the fly. And then when I was trying to ID this, I had never seen anything like this. And on a grainy FaceTime call to Richard Tehan and Connor Dooley, some cordyceps aficionados, they ID'd it as a hyperparasite. So there are fungi on the flies. Something to keep an eye out for. Arnie, are you in this chat and do you have the ability to speak?
don't think Arnie's here.
Okay. In our club, there is Arnie Martinson up in Bayfield, and he finds some cool cordyceps. Brennan, do you find any cordyceps in your outings down south?
Yeah, definitely. A few Cordyceps variabilis is what I find down here mostly. We found Stylifera in the northern part of the state as well.
Right on. Yeah. So the one that you can see in southern Wisconsin, if this was bright orange and a lot smaller and growing out of a log, Ophiocordyceps variabilis is one you can keep your eyes out for. And it really will just look like a teeny little thing poking out of a log, and you have to be very careful not to break it while you're trying to get the bug out to figure out what insect it's growing in association with. But there are plenty more than I can cover that you would be able to find in Wisconsin and around the Midwest. Here's one that you will not find in the Midwest because it has a relationship with insects that grow on bamboo. Ascopolyporus polychrois. They look like absurd, beautiful taffy balls. If you're hungry, it might make your mouth water. They look like candy. They're not. And they grow on whiteflies, aphids, and scale insects that are attacking bamboo. And so the story of how these grow is bizarre. We've heard of cordyceps taking over the bodies of an insect. What about if you're a fungus who just wants a good straw to suck on the bamboo and get the bamboo's juices? Ascopolyporus will grow and infect a scale insect or an aphid and destroy and consume the rest of the insect, but it will leave the stylus, the mouth part that is a straw, sucking on the bamboo, and it will use that to continue growing. Really bizarre life strategy, and there's a bunch of colors, and you can see varieties in the colors already here. This is one where I don't see why, if there's a big bamboo growth or plant or population somewhere, I don't see why there wouldn't be the Ascopolyporus. I've done work in bamboo forests in Hawaii that are all planted and introduced, and a lot of bamboo-specific fungi make it over. So the reasons that if you're vacationing somewhere and you're walking through a bamboo garden, you should keep your eyes out for a big ball, or a small ball rather, of taffy-looking thing sticking off of the bamboo. You might find yourself some Ascopolyporus slurping with another bug's straw. Nematode trapping fungi. So on our Earth, there has been a dance between fungi and nematodes for about 419 million years, is what we think. There are 4 different ways that insects or that nematodes are predated and attacked by fungi, and I'm going to talk to you about just one of them, nematode trapping. So these elaborate structures are made by fungi, which in a variety of ways catch nematodes. So this has evolved multiple times. And interestingly enough, the oyster mushroom family, so far as I know, all genera, all the different genuses in that family eat nematodes at some part of their life cycle. We know oyster mushrooms, Pleurotus, we know that some of them do it. We know Hohenboehlia, a relative, the shoehorn oyster, we know they do it. And there's a genus in that family in Africa that I can't recall the name of, but no one's looked to see if it does it as well. But it seems to be a popular way of making a living. And of course, we've got a reason for it: nitrogen. These nematodes are a good source of nitrogen for the fungus. So in this instance, we've got our Pleurotus tuberegium, and it makes a drop of neurotoxin. So these little drops, these are fungal hyphae, these are threads from this paper down here. And it makes a little drop of a neurotoxin. A nematode swims up, hits the neurotoxin, wham, bam, blam, game over. The fungus can immobilize it and then start to grow and consume it. Wild. But if drops of neurotoxins aren't enough for you, what about microscopic hula hoops? In the order Orbiliales, these innocent, they look so innocent. You'd never suspect they're vicious predators trying to trying to catch and capture nematodes. These little orbelia form hoops And when a nematode swims through that hoop, the hoop inflates and constricts around the nematode. And that's their way of capturing the nematodes, which is absolutely wild. There's a lot of detail and cool research about these lifestyle strategies. People have figured out when they kind of evolved these different structures. And as I mentioned, this is one of these ornate hoops and drops are just one of 4 different ways that fungi catch nematodes. Some of the others are pretty spooky, spores growing inside of the nematodes and whatnot. And I'm not a worm doctor, not an expert on worms, but I do know the nematodes that fungi catch are a specific type, and there are other types that fungi don't catch, so not all nematodes. So that kind of wraps up my talk for y'all. That's what I have to share. And I want to take a moment to recontextualize. If I had dropped you off on this slide and I said, thank you, good night, you might have been tempted to think, I just learned about a bunch of obscure fungi that, are kind of, it's cool to know about them, but yeah, they're just these weird one-off random things. I want to shift the narrative. Obscure fungi are the majority of fungi. Less than 1% of the fungi that we know form mushrooms. So that's the vast majority are ones in this pocket of obscure ways of living that we aren't thinking about. As many fungi, how many of the people on this earth are doctors? Just about maybe 1%, less than 1%. That's the same as the percent of fungi that form mushrooms. So if we're just thinking about them, we're really not doing our duty to study and investigate fungal biodiversity. About the same amount of redheads, right? 1% of the population. So we're only looking at the redheads? No. These are the kitchen staff. These are the ones holding down the fort. They're all over playing important roles in our world. And we're just greeted by the front end pretty gill-capped mushrooms. So they're important to think about. They're important to consider. And I hope that this gives you some tools to go out in your forests, wherever you live, and find something that you've inevitably walked by a dozen times. So with that, I want to point out a paper. If you're really loving it and you want to know 10 other unusual fungal niches, check out this paper by Sharon Cantrell. It's got cool yeasts and things in the Arctic and things inside of plant roots that we didn't touch on. So there's more for you if you're aching for more. And at this point, I can take any questions y'all have. Thanks for having me.
Yay! I just put the question in the chat, and yes, please ask your questions aloud.
I was
Go
just
ahead.
curious, with Lacnolula, and I mean, you can answer this more generally too. Is like the resin, the specific tree that is producing the resin, is that useful in identifying, or are they usually more generalist? And if you have other advice for IDing them down to the species, I'm curious as well.
So I know there's someone who knows a ton about this named James Danskin. If anyone can remember that James's name, who was at Harvard for a while, who knows about these, pop it in the chat for me. So I don't know the tons of details, but what I do know is within the family Pinaceae, we've got our hemlocks and some Doug firs and stuff, and our pines and such. There are some that only grow on some spruce. So I know there are some genera of resinicolous fungi restricted to some genera of trees. There's one called Lasiobolonium that is beautiful purple that I believe just grows on spruce. I have seen the same species on hemlock as I have on Douglas fir. So there's some jumping around. My educated guess is that there are going to be some that are more specific, and then there are going to be a little ones that are more general, but I don't really know.
Thank you. This is very interesting.
Yeah, thanks for listening. All right, I'm going to pop in the chat here and see amidst all of this gossip. If you read any, you can read them out to me, Mariah.
Yeah, I had seen a question that Todd asked. Todd Osmondson. So he asked, do Lichen fungi typically parasitize the thallus or the apothecia? And we were kind of having a little discussion that mostly we've seen both.
Yeah. I've read about ones that parasitize apothecia the most myself. Off the top of my head, I don't know if they parasitize the thallus much. I think the pink ones in our area are on the thalli of the lichens. So I guess both, but I'm
And I
not
think there's a lot
entirely
of little black dots, though I'm struggling sometimes to distinguish when the little black dots are like a Nicholas fungus or they're just like a feature of the
Yeah, that's a challenge. And you can scope things to be more confident, but also it's just tricky. Some lichens, as they degrade, turn funky colors. So it's hard to be feel confident about that. I'm seeing Todd point out some alpine fungi. So there is Rhizomerasmius epidryas. There's a little mushroom that only grows with a species of Rosaceae, I believe, up in the alpine. There are fungi that we only really see, above the tree line up in the mountains too. That's a very unusual and specific niche, and one that I'd love to explore more.
Yeah, there's plenty of fungi that's on thallus of lichens. Actually, those are usually slightly bigger and could be seen with the naked eye usually, as opposed to ones on apothecia. They're so tiny that you need a microscope or good glasses. So the ones on thalli are usually giant compared with the ones on apothecia.
That's good to know. That's helpful, yeah. Ones on the thalli are easier to see. Let's see. Todd says when you ignore Tabaria, you could be ignoring an entire ecosystem. I think we're in a group of people who are paying attention to all the little brown mushrooms. Kiona found some dryad saddle and they were covered in something the next day before she could eat them. That's interesting. Yeah, they're a microhabitat. I would love someone to just do a study of everything that grows in association with old dryad saddle. Would be beautiful.
Any other questioners want to unmute yourself or type it in the chat and we'll read it aloud. I have some questions.
Oh yeah. Thank you for
Nobody
finding
else
James
does.
Cameron Mitchell.
Oh yeah.
Yeah, he's someone who knows, or they are someone who knows a lot about resinicolous fungi. If you're interested, someone you could bother by emailing them and maybe they'd answer.
Yeah. I trying to remember some of my questions. I had a lot of them along the way, but then you try to kind of pop them in the chat. One of them was the Mycobacterium. I was just trying to find that online, and I was having a hard time. It kept trying to tell me about Mycobacterium. So I was just kind of little.
Maybe Mycobacterium stellariae using an epithet could help get something to come up.
Cool.
There's
Good
a
idea. I see. I'm spelling it wrong. It's micro.
Oh, it is micro?
Yeah.
Botrium. Oh, so did I misspell it? Well, that'd be
Well, could have been me, could have been you, but it's very, very easy to do. There's a lot of micros and a lot of mycos. So
There's
I'm putting it there. Great.
if you're a botanist or you have botanical friends and you want to give them the instructions to try and find that in your region on chickweed, there's a recent review paper on that genus, and if you download the supplemental files, it has a big list of all of the known plant hosts of that genus. So that's one if you're really going for it, that's a way to find the hosts near you that could potentially have it.
Find a Mycobacterium host near you.
Yeah, that's right. All right.
Yeah. And I'm assuming that people can also direct their questions that come to them in the middle of the night to Jack at his Gmail there, or reach out to him on iNaturalist or Instagram.
Yeah, feel free. And if I don't know too much about it, I'll point you to the person who does.
Very cool. Honestly, Jack, one of the most action-packed talks we've had. Really cool. And I want to leave everybody saying that, like, I want to see these things coming in on the iNaturalist project for us. Get out there, see them. Same for that. If you recall, when Fenner spoke to us, I pointed out Xylaria tatacula, I think, the little fairy sparkler. We're going to find it. It's surrounding Wisconsin. It's here. Don't let the Midwest laugh at us anymore. We also have the fairy sparkler. And yeah, Jay Justice and Ananda are thanking you in the chat, and I thank you too, and on behalf of the whole Wisconsin Mycological Society. So awesome talk.
Thanks for having me. It's a great club to be a part of.
And we have recorded this one, so it can go up on YouTube, and you can share it with all your friends and get them to find a really cool weird fungus too.
Excellent.
All right. Good night, everyone.